Researchers have conducted the most thorough study of acute flaccid paralysis incidence over the past few years in the United States. Welcome to Microbial Minutes, ASM's update on what's hot in the microbial sciences, the What's Up with AFM edition. I'm Julie Wolf, Science Communications Specialist here at ASM, and today we'll be talking about a paper recently published in Pediatrics that provides a long-term study of the officially reported cases of AFM. Now, a rise in acute flaccid myelitis, or AFM, first made the news in 2014 when several children in Colorado were identified as cases of flaccid paralysis. They had the inability to move their arms and their legs, and this unusual syndrome was reminiscent of polio, another disease which is also caused by a viral infection, or one that is caused by a viral infection and ends up with flaccid paralysis in a small number of infected patients. In response to the biannual outbreak of this AFM mysterious condition, the CDC developed an investigation unit around AFM, as well as better defining the cases and a reporting protocol. Now, this isn't a nationally reportable disease, but many states do require that incidences of AFM be reported, at least at the state level. Finding a causative agent of AFM has proven difficult because many of the samples that have been tested show no virus or other potential microorganism that might be associated with this output or with this outcome. And what I'm showing you here is the symptoms of acute flaccid myelitis. This is something that is delivered to parents because to keep a watch over their children, since most of the symptoms have occurred in young children, as we'll discuss. And although we will be talking about cases that were reported between 2015 and 2017, this 2018 reported case map shows you that these cases are reported really broadly across the United States. So this is not locally concentrated within one region within the U.S. Now, in the pediatric study, the research team wanted to look at AFM in a more unbiased manner. As the definition was changed from focal paralysis to flaccid paralysis, experts from the CDC and U.S. academic institutions started to classify the patients as confirmed or probable cases based on reviewing the patient reports. Sterile sites from patients were collected, things such as blood, serum, and cerebrospinal fluid, or CSF, as well as non-sterile sites, such as the oral or respiratory samples or stool samples. This data was separated into cases that were confirmed by the CDC laboratories or non-CDC sites. And first we'll look at the sites that were, the samples that were confirmed by the CDC labs on the next slide here. What did they find? So if we look at this table, the very top numbers are the numbers of samples tested, and the columns over here on the left-hand side represent the different viruses that they looked at in those different types of samples. So as CSF and serum or plasma were tested as the sterile site representatives, and the respiratory or stool swabs were, stool samples or rectal swabs were the non-sterile sites that they looked at. In this table, you can see that many of the positive samples were identified in those non-sterile respiratory and stool samples over on the right-hand side, but almost no samples were positive in the sterile samples. Among the CDC tested samples, 1 CSF and 2 sera samples were positive for some sort of virus, and that was among 81 and 72 different samples tested respectively. Now, on the next slide, we'll see that in the non-CDC tested samples, 4 CSF and 2 sera samples were positive for viral presence, and that's among a larger cohort of 170 or 123 samples tested respectively. Overall, from 2015 through 2017, 43 states reported 305 cases of AFM, and 193 of these were confirmed by the CDC. 61% of the patients were male, 53% were white, and 35% were non-Hispanic. The median age of onset was 6 years, so this is a condition that often affects very young children, but not entirely. Of the 161 patients who showed respiratory illness, there was 1 death reported, and that occurred in a 21-year-old patient. So the age range is not restricted to only the very young children. Of 178 patients who were administered an MRI, 80% showed spinal lesions and 65 had brainstem lesions. Importantly, all of the specimens were looked at for poliovirus and none of them tested positive. So this is definitely not an incidence due to poliovirus infection. Now, among those non-sterile specimens, Enterovirus D68, one of the leading suspects as a causative agent of AFM, was detected in similar levels to the cases that had been collected in that initial 2014 outbreak. But EVD68 was also found in patients later determined not to have actual AFM. So they were cases that were not included in the confirmed cases among the CDC confirmed cases. There is a specific assay for Enterovirus D68, which allows researchers to specifically probe for this particular enterovirus among all the different enterovirus strains, but this may be limiting the detection of other circulating enteroviruses that could contribute to AFM. Enteroviruses are ubiquitously circulating viruses that have seropositivity or seroprevalence depending on the strain, in 33 to up to 99% of the population. So these are very ubiquitously found and infecting viruses. Most of these enteroviruses cause respiratory disease that does resolve without further sequelae. The authors suggest that this Enterovirus D68 is still a suspect based on the positivity in many of the samples. But because many people have been infected with this Enterovirus D68 but don't come down with AFM, other factors must be playing a role in development of this very rare outcome. And they suggest things such as host genetics or co-infection with multiple pathogens. Studies in mice have demonstrated that EVD68 can cause neurological disease in mice, and we've covered the serological evidence of previous EVD68 infection in AFM patients on a previous Microbial Minutes. The study was subject of a commentary, which was also published in Pediatrics, and we'll see that on the next slide. In the commentary, the authors emphasize that this particular primary literature does confirm the biannual periodicity of AFM cases, and we can see that periodicity in the graph here. The blue boxed regions are those CDC confirmed cases, and the red box here outlines the cases that were looked at in this study. Most reports in the pediatric study were made between August and October of 2016, but the reason for this pattern, this August and October prevalence every other year, remains unknown and one of the biggest mysteries surrounding AFM incidence. Now, we'll link to both that commentary and the original Pediatrics report, as well as our previous Microbial Minutes on EVD68 antibodies from AFM patients. Now, to learn more about Enterovirus D68, you can listen to a recent This Week in Virology podcast episode, which we will also link, and there's a video associated with that as well. Today, we've heard that the largest systematic study of AFM continues to point to a viral etiology, possibly Enterovirus D68, but without strong causative evidence yet in hand. For more enterovirus and AFM updates, be sure to subscribe and ring the bell for notifications every time we add a new research news video. I'd like to thank you for listening and thank Ray Ortega for production. I'm Julie Wolf, and I'll be with you next time on Microbial Minutes.